Hello, my name is Richard White from the London School of Hygiene and Tropical Medicine, and I'm going to talk to you about using epidemiological modelling to estimate TB vaccine health and economic impact, and then my colleague, Dr. Sophie Rhodes, is going to follow. um, we tend to use mathematical modelling to understand how different vaccine characteristics will affect population health and economic impact. And we use the mathematical modelling as the kind of framework to pull all those various disparate bits of information together and try and make a prediction. And we do that for funders who typically are putting together target product profiles or indeed preferred product characteristics such as WHO. And we pull together, once we've got it set up, we can pull in new data, be it clinical trial data or new epidemiological data, and update those potential estimates of impact to inform their decision making. And the idea is that in the context of a kind of very busy vaccine pipeline, mathematical modelling can continuously incorporate those new data and support and course correction of TB vaccine development. So that's kind of what we do. I'm not going to talk about anything in great detail because you're not really interested in the details of the results, but I'm going to talk more about the methods because I think that's probably what you're more interested in, and just give you a couple of examples of the sort of things that we tend to be able to provide to decision makers. So we tend to use age-structured compartmental dynamic transmission models to capture the indirect effect of these interventions, given these are infectious diseases. And we quite often use age-dependent social contact mixing patterns because that's important for understanding the spread and implication of vaccine age-specific targeting on these vaccines. We set up an ordinary differential equation model, either at discrete or continuous time. There's a few boxes you can see, so it can be susceptible, you can be latent, you can be infectious boxes and recover boxes. And the arrows moving people to and fro those boxes. And obviously we will then stratify that natural history model by other really important aspects of the modelling, which is vaccination, whether you're vaccinated or not, and those kind of rates of flow and likelihood of becoming diseased or dying are going to change as you're vaccinating. And then there may be other important characteristics. It's not just about TB, so in high HIV endemic countries, HIV, all those compartments will be stratified by HIV as well. So you can imagine it gets reasonably complex reasonably quickly. In essence, there are methods to basically calibrate a baseline scenario or no vaccine scenario, and that incorporates all the important things about the bug, which you've just seen, but also the kind of health systems and the ongoing control strategies or work that's going on. So there'll be a certain level of case detection or estimated case detection within a country, and we model that ongoing non-vaccine interventions in the baseline scenario. And some countries have very different health system setups. So there might be a very, very strong private sector care in India, so we incorporate that explicitly. We incorporate the fact that China's got very high levels of case detection. And for example, in South Africa, we have to model HIV and ART trends as well as those unfortunate epidemics have hit that country. And the interventions to stop it have been rolled out. We then take that kind of model of the health system and the epidemiology, and we calibrate to some key aspects of the population. It will probably be TB burden by age, potentially over time, and it varies a lot on kind of the availability of the data. Some countries have richer data sets than others. We try and do that as statistically robustly as possible, using something random like Latin hypercube sampling with a set reject on those targets. And then we might pull in a couple of those targets if we're running out of time near a deadline using ABC, approximate Bayesian computation, or MCMC. We've more recently been experimenting with emulation and history matching, which I'll be very keen to talk about because that seems to be very promising actually. But the idea is you get about kind of 1,000 or so or 10,000 parameter sets, which incorporate the uncertainty and the things you don't know, those priors, but then always all fit all those calibration targets. So it kind of looks like that country that you are meant to be representing in your model pre-vaccine, the new vaccine. So we have a nice population, the model that looks like that population as much as we can, and then we think typically model different vaccination characteristics. So for example, here, whether the vaccine protects against infection or disease. That's quite an important characteristic. Whether it works in infected or uninfected individuals, whether how long the duration of their efficacy works, how high the coverage is, who you're targeting, infants, middle-aged people, old people, and whether those characteristics change by HIV status of the individual. Going into a bit more detail about some of those, just to understand TB, to understand some of the results later on, whether it works, whether it protects against infection or disease. So that, this is again is our TB kind of natural history curve with uninfected, latently infected, diseased individuals, etc. And a prevention of infection vaccine will block some of those pathways, so from getting to infected or latent or recovered post-disease, post-treatment through to latently infected again. Prevention of disease vaccine won't stop you getting infected, but does stop progression to disease either from the uninfected state or the latently infected state, or indeed from relapsing from the recovered state. So, and that will have a different impact depending on the numbers of people in those various classes in different countries, which will vary. Thinking about pre versus post-infection, basically whether the vaccine works in uninfected or infected individuals, again, that same kind of natural history curve, then a potential infection vaccine will be given to everybody, but will only be effective in uninfected individuals. And similarly, a post-infection vaccine will probably be given to everybody, but would only work in latently infected or recovered individuals, individuals who have already been infected. And that has depending on how high infection prevalence is in that country, that will obviously vary a lot. The numbers of people who get protection will vary by epidemiological setting as well. Okay, so we take those calibrated data sets and we take those bunch of vaccine characteristics that we want to model, and we might plot something like this with incidence rate over time. So basically what we think will happen if the vaccine is not introduced. And as you can see rather dismally in TB incidence rates are going down at about 1 or 2% per year, very, very slowly in the absence of new interventions, new stuff. We introduce the vaccine and TB incidence goes down, and we might show that as an impact on TB incidence in 2050. You can see here the vaccine of a certain efficacy against infection and a certain efficacy against disease had a certain population level impact, which is the blue star, and a different vaccine with a higher level of protection against infection and a higher level of infection against disease has a higher population level impact. So we use those heat maps to try and illustrate clearly what the impact of those different vaccine characteristics would be in these model populations. Okay, so what do we see for that? I'm just going to focus on age targeting as my first result. There's lots of things I could show you, but that's not the point. I'm just going to show you a couple of key results. So here it's age targeting, whether we target infants or adults and adolescents first. And this was a result that was quite influential in around about 2014 when we first showed it, that showing if we target neonates, which is typically how BCV, our current vaccine, is targeted right now. It's targeted typically at infants, neonates. If we introduce another vaccine like that, we're going to get very little impact for a very, very long time. This is 2050. And that's because most of the disease, TB burden globally right now is in adults, and that's where the infectious disease is as well. That's infectious pulmonary TB, basically. So if you vaccinate kids, just demographically, you're going to have to wait 20 years before those kids, 15, 20 years before those adults grow up and start coming into the age groups where they have the sorts of disease which is most important globally. Whereas if you're able to get a vaccine that we can use in adults or adolescents, you can see you get a much greater impact much quicker. This is when the vaccine is introduced, whereas nothing for 15, 20 years, very large reductions in burden before 2050. So just purely demographically, or the interaction between demography, vaccine age targeting, and the TB epidemiology, a vaccine that was able to work in adults or adolescents and be delivered to adults or adolescents would have much higher impact. And then secondly, I just want to show what the differences in characteristics, what the importance of different, whether it works against infection or disease, and whether it works in infected or uninfected individual has on potential impact over the next 20, 30 years. Here's a scenario we model. So it works in prevention of infection. It prevents infection and disease. We'll model a wide range of vaccine efficacies. Because we don't know what the eventual vaccine efficacy will be, we model a wide range in durations of efficacy because we don't know what the eventual duration of efficacy would be. We model it in 3 populations because these are kind of widely and kind of well-known different epidemiologies. So they're quite representative populations globally. We model it as roughly as effective in HIV positive individuals. We model we varied whether it worked in uninfected or infected individuals or both. We rolled it out in 2025. We gave it, remembering the recommendation from earlier to focus on adolescent or adult populations, we rolled it out routinely to 9-year-olds and in mass campaigns to 10+-year-olds. And we modeled in this one-off scenario I'm showing here quite high levels of coverage. And this is typically what you get, a kind of set of heat maps like this, showing impact in colours, vaccine efficacy against infection against the x-axis, and vaccine efficacy against disease on the y-axis. This assumes, this particular scenario I'm showing you here, works in both pre and post-infection, so it works in everybody this vaccine. Those 3 populations, again, low transmission, aging epidemic in China, high transmission, younger epidemics in South Africa and India, against disease on the Y, against infection on the X. And what this, in huge summary, what this shows, these heat maps show, is that protection against disease provided greater impact regardless of setting. So getting a vaccine that protects against disease rather than protect against infection gives you greater impact before 2050. But saying that, in the settings with high levels of transmission, although protection against infection didn't give you higher impact than protection of disease, it became more useful. And that's kind of intuitive, isn't it? The more ongoing transmission is there, the more protection against infection vaccines would contribute. We'd have a bunch of those results. We try and translate them for the decision makers. So here is just an example about what we think it means for a recruit if you're trying to run a trial, who should be recruiting. So if maximizing population level impact by 2050 is your goal, and it doesn't have to be, of course, but if it is, then development of vaccines for adults and adolescents should be prioritized based on our results. You should be recruiting post-infection or already infected individuals. But that said, that in populations with higher transmission, such as India and South Africa, the pre-infection populations could also be recruited. And ideally, if you have the money, we'd it'd be good to assess efficacy in both those populations. We'll then obviously try and summarize it all right down for our audience and make some statements that hopefully resonate with them, such as the impact will differ by epi or health system setting, and we've hopefully showed them how it might differ. And might some takeaways such as one size probably won't fit all, and one vaccine type won't like to fit the global 2050 TB goals, and therefore more than one new vaccine are likely to be required. But that said, the new and repurposed TB vaccines currently in development could provide an important and cost-effective reduction in global TB burden. So kind of message here is to keep the pipeline diverse, but the vaccines already in late stage are definitely worth taking forward because they could be effective. It's a continuous bit of work, massive team of people. These are external collaborators to us, but there's always also a very large number of people internally who are working on this. I'm very keen to get your questions. See you in a bit.
Hi, so I'm going to do the second part of the presentation from LSHTM entitled Using Vaccine Immunostimulation Immunodynamic Modelling Methods to Inform Vaccine Dose Decision Making. So just as a background, mathematical modelling is used extensively to guide drug development. Obviously, there's PK/PD modelling, and drug dosing being one of the main things that's determined using this modelling method. However, vaccine development does not benefit from the same level of quantitative analysis. Vaccine doses are selected based purely on the empirical data that come out of the trials, which sometimes can be limited, which is understandable because testing many doses can be quite resource heavy. In addition to this, there's little preclinical to clinical dose response translation. So not much data is taken from animals to humans to try and predict the dose response curve in humans. There are many detailed data coming out on vaccine immunogenicity and efficacy these days. So we think that that's a very exciting opportunity to apply the same sort of quantitative methods in vaccine development that have been used in drug development. So our overall aim is to use these modelling methods, so PK/PD type modelling, to accelerate vaccine development. So as a definition, we've created a new term called immunostimulation immunodynamic modelling, ISID. And this is the field that we're proposing to address the lack of quantitative methods used to optimize vaccines. Now, immunostimulation immunodynamic modelling is a new term, but it sort of breaks down as follows. Immunostimulation is very much the immune response that's stimulated following vaccination, and immunodynamic modelling is or the immunodynamics is the measured immune response that we're interested in modelling. So whether this be the T cell response or the antibody response. We created this term to reflect that we were trying to use PK/PD type modelling, but not necessarily the same type of dynamics, because we won't necessarily be dealing with the same type of data. So one of the first examples of ISID modelling we've been working on is in TB vaccines. As Richard mentioned, we're the TB modelling group. We've done a lot of work on the epi side of TB vaccines, and this is the sort of first example of ISID in the TB vaccine space. The idea is to take the animal dose response curve and translate it to the human dose response space. Now, we do this by combination of mathematical modelling and also dose allometry, which I will explain both of those things in a moment. But the idea is to use the animal dose response, which you can see in this quite simple picture on the left-hand side, to tell us where we should start testing vaccine doses in humans, and just give us a range of the best dose. And just as a quick note, when I say dose, I mean antigen amount in this case. So the aim of this piece of work was to use ISID modelling calibrated to longitudinal TB vaccine, and the vaccine in question is called H56, which is a subunit vaccine. Multi-dose response data in mice to predict the human H56 dose-dependent response dynamics. Now, we split this into two steps. The first step was to calibrate a mathematical model to the longitudinal response data in mice and predict the full dose response range at a late time point. And then the second stage was to map the dose response in mice to the human dose response space. So just to give you an overview of the data we were using, we conducted a multi-dose, multi-time point experiment of this novel subunit TB vaccine H56, which at the time was adjuvanted with IC31. And this was done in F1 mice. We tested 6 doses, ranging from 0.1 micrograms of the vaccine to 15 micrograms of the vaccine. We kept the adjuvant dose constant throughout, and we also had a control data set. The mice were given 2 vaccinations, one at day 0, one at day 15, so they were boosted at day 15, and this was to mimic the human trial, which I'll show you in a second. And importantly, we actually grouped some of the dosing data together to give us more power with the modelling. So we grouped the 0.1 to 1 micrograms, and we call this the low dose group, which we averaged to 0.5. the middle dose group, which was 5 micrograms, and the high dose group, which was just the 15 micrograms, which helped us with the scaling properties that we needed to go from mouse to human, which I'll explain again in a second. And then in order to make the translation from mouse to human, we also needed some human data. So what I show you here is actually a pooled data set from H56, the vaccine we were using in mice, and H1, which was actually its predecessor. But because they're quite similar immunogenicity, we felt that we could pool the data to give us some more power. So this is actually from a phase 2A trial in South Africa. All participants were HIV negative and non-latently infected, and there were 16 in total. But importantly, the dose they were given was 50 micrograms of the vaccine, and then the adjuvant dose was slightly more than we gave to the mice, but it was kept constant as well. So that's the human data set. Now, the model that we used was designed specifically to represent the data, as the data is interferon gamma secreting CD4 T cells, which at the time was thought to be an important marker for protection in TB. We actually don't quite know exactly what is protective in TB, but this is the best option that we have at the moment, or when we did the study. So the model itself is quite simple. It's two compartmental ODE model. The two compartments are transitional effector memory cells and resting central memory cells. When the model starts, the cells are recruited straight into the effector memory compartment. They either then die or they move on to resting central memory compartment, and that's where they'll stay until they get until the boost vaccination. At the time of boost, the central memory cells actually replicate for a certain amount of time, and then they move back into the effector memory compartment, and then the whole process will continue on from there. So although it's only two compartments, the dynamics are actually fairly complicated because of the effect of the boost vaccination on the interferon gamma secreting CD4 T cells. So that's a very simple description of the model. That's what we were going to fit to the data. So I will now show you the results of doing that. So we calibrated the model to the data, as you can see here. I won't explain all of the points on this on these graphs, but just to say that the blue line and the blue triangles, the blue line being the model prediction and the blue triangles being the median of the response data, line up quite well. So that means that the model has done a good job of predicting the median response data. On the left-hand side plot, you can see the low dose group data in mice. The middle plot is the middle dose group, and the right-hand plot is the high dose group. So you can see that throughout the model is doing a good job of predicting the data, which is the first sort of tick on our checklist. I've circled the latest time points because that's where we'll be focusing the translation. But as you can see throughout, over the whole time scale, the model's capturing the response well. So that's good. And this is the same results for the human data. The model again does a good job. The data is a bit more variable, but it shows the sort of average trend quite well. And I've circled the latest time point here as well. So the second part of this work was then to map the dose response curve from mice to humans to try and make predictions for the human dose response space. So here you can see a dose response plot using the mouse data. Now, instead of this being the x-axis of on time, this is now dose. So it's slightly different. The triangles represent the interferon gamma responses. So the grey triangles are all the individual responses, and the red ones are the median. The blue points are the calibrated model prediction for the median, and then the orange circles are then model predicted interferon gamma responses for intermediate doses not actually tested in the experiment. The star represents the most immunogenic dose, and the green lines are the range of that, because we grouped some of the data together, if you remember from the data that I showed you. So that's the mouse data. That's the dose response curve at a late time point for mice. So now we have to think about how we actually translate across. So to map the dose-dependent responses, we need a dose equivalency across the species, and I'm calling this the dose allometric scaling. In the case of H56, we actually assume that the dose allometric scaling factor is 10. So this is the current published sort of assumption around these H series vaccines in TB. So in the case of this data, this means that for the 50 microgram dose in humans, we're saying that the equivalent dose group is the 5 microgram dose in mice, which was the middle dose group. Again, adjuvant dose kept constant here. Therefore, what we can say is that the remaining predicted human doses that we don't have data on are just 10 times the mouse dose for this particular case, because the model was actually quite simple when we calibrated it. But when we did this work, we did a sensitivity analysis around the scaling factor, informed by a literature search, but we're just showing the results for the scaling factor of 10 here. So using that scaling factor of 10, we actually predict the dose response curve on the right-hand side for humans. So where I've drawn a box around the 5 microgram dose and the 50 microgram dose, 5 in mice and 50 in humans, that's how we sort of anchored the two together. And then we just predicted the dose response curve across from mice to humans using that scaling factor of 10. Again, because the model output was actually quite simple, this is just a case of scaling everything up by 10. But given in more data, more complex data, this might not be as simple. But in this case, it was quite nicely sort of a nice prediction. So in total, we found that the human range of most immunogenic dose was actually between 1 and 10 micrograms as predicted by the model. As I just said, the modeling predicts a lower dose. Lower doses may be more immunogenic in humans based on the mouse data, then say the higher doses actually gave less immunogenicity than the middle doses, which is sort of another topic altogether, why the dose response curve looks like that, which I'm happy to answer questions on. But for the moment, that's kind of, that's where we predicted our most immunogenic dose would be. And actually, these model predictions were validated by an empirical trial that was running parallel to the work that we did, where in in humans, the H56 vaccine showed that a lower dose, which was in between that range, was shown to be as immunogenic as higher doses. Now, I don't think this trial was powered really to see a difference between doses, but they kind of agreed with the results that we predicted, that the lower doses would be the one to go for, and that's what's been chosen for the rest of the development for this vaccine. So in the last few minutes, I'll just talk about our current and our future work, mostly just the future work actually. So there's a lot of applications for ISID modeling. I've only just shown you one, which was the cross species translation, but there's many other things we can do. So for example, timing of boost, so regimen of vaccine, when should we boost, how much dose should we give when we boost, and how many boosts should we give. This can all be simulated. Adjuvant and antigen dose, obviously I only talked about antigen dose, but the adjuvant's very important too, obviously, so we need to think about the dose for that. Optimal design is something that is very interesting. So how can we use our model to design more efficient trials using the resources that we have? So we want to get the most information out of the least possible resources. So optimal design is something that I think could really work very well for vaccine development. And then kind of on the other side of that, making a software so that these simple models, or simpler models than the one I presented perhaps, can be used by vaccine developers, so integrated straight into development, as opposed to having to talk to a modeler and having to design a brand new model. These sort of very simple ways of summarizing the dose response curve should be made available to people. So that's something else that we would like to do in the future. So as a summary, there is, I wouldn't say no use of quantitative methods in dose finding in vaccine development these days, but there is obviously quite little compared to drug development. And we think that ISID modelling is the answer to that. And we really think it could accelerate vaccine development if it's used in the right way and communicated in the right way. We've used ISID modelling on novel TB vaccines to translate dose response curves from mouse to humans, but there are many, many other applications of ISID that I think are definitely worth exploring and have shown in drug development to be vital to make really important decisions for drugs. We should be doing the same for vaccines. So I'd just like to give some acknowledgement. There was a lot of lab work that went into finding getting the mouse data, and we had a great team at LSHM who did that a few years ago now, and they worked really hard, and it was quite intensive, but it was really great. The funding for the project came from ERAS, which was formerly TB vaccine developer, and the Gates Foundation. We had input from the vaccine developer, SSI, and fellow TB scientists and fellow PK/PD modelers. So thank you very much for contributing to that work. And if you would like to know any more, please email me or please ask me questions. And I really appreciate your time. So thank you very much for listening.